there is a vascular layer that is known as tunica vasculosa, and from this fibrous layer some septas are introducing into the substance of the testis, and these septas are dividing the testis into some lobules. Each of the lobules contains 2 to 3 seminiferous tubules. Okay, these seminiferous tubules, they helps in formation of the sperms, helps in spermatogenesis. Okay, it is lined by the stratified epithelium, seminiferous epithelium. This is a special type of epithelium. So, and the end part of the seminiferous tubule is straight. These are known as straight tubules. These straight tubules join with each other and form a meshwork. This meshwork is known as the retitestis. Okay, and from retitestis, these are the efferent ductules. They enter into the head of the epididymis. From the head, the sperm goes to the body and tail part of the epididymis, then passes through the vas deferens. Okay, and this testis is covered by a peritoneal sac. Okay, it was previously connected with the peritoneal cavity of the abdomen. This is known as tunica vaginalis. During the descent of the testis, some part of this peritoneal lining is coming down with the testis. So this part remains patent. This is known as tunica vaginalis. In hydrocele, there will be fluid collection within this tunica vaginalis. Okay. So the testes are, testes for single you use TES, TIS, and for double TES, TES. Okay. So that is the difference. So testes are paired ovoid masses or organs that lies in the sac of scrotum. Testis develops from intermediate mesoderm of the genital ridges in the abdomen and migrated primordial germ cells of the yolk sac. Later the testis descends to lie in the scrotum. Okay. These are the structures of the testis. Outermost fibrous covering is known as tunica albuginea, tunica vasculosa. Then from the connective tissue, there is connective tissue septa dividing the testis into lobules. There is mediastinum testis formed by the meshwork within which lies the retitestis. And there are seminiferous tubules within the lobules, and seminiferous, in between the tubules there are some interstitial cells of Leydig which helps in secretion of the testosterone. Last part of the seminiferous tubule is known as straight tubule or tubuli recti, and from the retitestis arises the efferent ductules enter the head of the epididymis. Okay, so that structure we have already discussed. Next we go to the seminiferous epithelium. So seminiferous epithelium, this is a specialized type of epithelium. Seminiferous tubules are lined by complex stratified epithelium called the seminiferous epithelium, and it consists of two types of cells. One is supporting cells, these are known as Sertoli cells. Another is spermatogenic cells. Okay, these two types of cells are lying within seminiferous epithelium, and each seminiferous tubule is approximately 50 centimeter long and 150 to 200 micron in diameter. Okay. See here, like this you will see under the microscope. Outer covering is tunica albuginea. Then there is tunica vasculosa. Okay. And these are the sections of the seminiferous tubule lined by the seminiferous epithelium. You can see multiple dots for different layers of cells, spermatogonia, different types of spermatogonia, then primary and secondary spermatocytes, spermatids, like this. And this is the lumen. Okay, near the lumen lies the sperms. And after from the seminiferous tubule, they will enter into the retitestis, then via efferent ductules into the epididymis. From there, through the vas deferens, it comes out. Okay. So this is high power view. Previous one was low power view. Like that view you will see under the microscope. So this is high power view. Here within the lumen you see the spermatids that will transform into the sperm, and these are this large nuclei. These large nuclei are of the Sertoli cells, and this dot-like nucleus, all these are for the Spermatogonia lying at the basal layer. Then they are going through mitosis, forming the primary spermatocyte. They are dividing and forming the secondary spermatocytes, and then they are forming the spermatids, and from there sperms are developing. Okay. So we'll discuss individual cells in details. Sertoli cells or sustentacular cells. This is individual short note. Sertoli cell, important short note. Okay. Sertoli cells or sustentacular cells, these are supporting cells. They are columnar or pyramidal in shape with light staining euchromatic nucleus. On HE staining, these cells cannot be distinguished. Sertoli cells represent true epithelium of the seminiferous tubules, and there are Charcot-Boucher inclusion bodies are present within the cells. These are the functions of Sertoli cells: secretion of androgen binding proteins ABP, secretion of inhibin, secretion of plasminogen activator, and transferrin. Transferrin is iron transporting protein. They support and nourishes the germ cells, phagocytosis of the residual bodies after the spermiogenesis. So all these are functions of the Sertoli cells. Next we go to the germ cells or spermatogenic cells. These are derived from primordial germ cells. They replicate and form various stages of spermatogenesis. They are arranged in stratified manner from basal lamina to the luminal area as follows. First Spermatogonia, then develop into spermatocytes, then spermatid and spermatozoa. In any one cross section of seminiferous tubules, all these cells may not be seen. Spermatogenic cycle, it is a wave of germ cell maturation that reverses along the length of the seminiferous tubule. Next we go to the lamina propria or tunica propria. It is a thin layer of vascular connective tissue that surrounds each seminiferous tubule. That is tunica vasculosa. Okay, lamina propria represent the tunica vasculosa. Lamina propria do not contain fibroblast. It consists of 5 layers of contractile myoid or peritubulous cells external to the basal lamina of the seminiferous tubules. Rhythmic contractions of myoid cells help in the expulsion of sperms and seminiferal tubule secretions. Okay. So next we go to the Leydig cell or interstitial cells of Leydig. This can also come as an individual short note. Okay, Leydig cell. So Sertoli cell, Leydig cells are important short note. Leydig cells lie in the connective tissue between the adjacent seminiferous tubules. They are large polygonal eosinophilic cells with round nucleus. Leydig cells have LH hormone receptors. LH stimulates Leydig cell for production of the testosterone. Steroid secreting foamy eosinophilic cytoplasm because Leydig cells have well developed smooth endoplasmic reticulum. What is the function of Leydig cell? Testosterone synthesis. And there are crystals of Ranke within the Leydig cells. Next we go to the process of spermatogenesis. This comes under embryology long question. Okay, it can come as a long question or part of the long question. Spermatogenesis. Spermatogenesis is formation of spermatozoa from the Spermatogonia. Spermatogenesis occurs in 3 phases: spermatocytosis, meiosis, and then spermiogenesis. All these terms you remember very carefully. What is the process? Okay, All this can come in short questions or can be asked in your viva, what is the process. So spermatocytosis is conversion of spermatogonia into primary spermatocyte. Human spermatogonia are classified into three types based on the appearance of nuclei in H staining. These are as follows: type A dark spermatogonia, type A pale spermatogonia, type A spermatogonia, and type B spermatogonia. All are different types of spermatogonia. Then spermatogonia undergoes meiosis and develops spermatocyte. So meiosis, it involves the conversion of primary spermatocytes into spermatids, and primary spermatocyte on meiosis form two secondary spermatocytes, and that later undergoes meiosis 2 to produce 4 spermatids. Okay. So 146XY cell, after the meiosis division, it will form this having genotype this 23X, 23Y, and after the second meiosis division, it will develop into 4 cells having these genotypes 223X, 223Y, and each spermatid contains single stranded haploid DNA. Okay, 1N. In diploid we have 2N, so in haploid we have 1N. Next we go to spermiogenesis or spermatid phase, third phase. Spermiogenesis is process of metamorphosis of spermatids by which they get converted into a spermatozoon. And the metamorphosis is the change in the form of the cell. 4 phases of spermiogenesis are these. First is the Golgi phase, cap phase, acrosome phase, and maturation phase. Most of the cytoplasm of the spermatids is said as Raynaud's residual body, and Sertoli cells phagocytose this residual body. Okay, this is one important function of the Sertoli cell for metamorphosis of the spermatids into spermatozoon. Next is permeation. This term means you very carefully remember which process is included in this term. So spermiation is finally spermatozoa are released into the lumen of the seminiferous tubule, and this is known as spermiation. Okay, only the release process is known as spermiation, and metamorphosis is known as spermiogenesis. Okay. Next we go to structure of mature sperm. This can come as a short note. You also practice a schematic diagram of a mature sperm. Okay. So mature spermatozoa have a head, neck, middle piece and a tail. Length of the sperm, mature sperm, is about 50 to 60 micrometer, and in count of the sperm in single ejaculum there is about 200 to 300 million sperms are emitted in a volume of 2.5 ml of semen. Head of the sperm. So we will discuss individual parts of the sperm now. So head of the sperm, it is pyriform, that is flattened and pointed, and the acrosomal cap covers the anterior 2/3 of the nucleus. Acrosome contains digestive enzymes. These are hyaluronidase and acrosin, and that help to break the outer wall of the ovum. Okay. Next we go to the tail. After the head, mainly it has 2 parts. All those parts, neck, middle piece, all those are included within the tail. Okay. So the tail of the sperm is divided into neck, middle piece and end piece. Okay. Neck, middle piece, annulus. Annulus is ring-like structure. There is a principal piece and axial filament, axonemal complex, that forms the end part of the tail. So maturation and capacitation of the spermatozoa. Sperms released from the Sertoli cells are non-motile, and they get partial motility in the epididymis. So this process is known as the maturation. And capacitation, these terms are also important. Maturation of sperm, capacitation of sperm. Capacitation, after ejaculation, the sperms in the female reproductive tract undergoes capacitation. It is the maturation of sperm and it involves removal of the glycoprotein coat from the sperm, and it enhances sperm motility and makes the acrosome ready to release lysosomal enzymes, okay, for penetration of the ovum. So see here different cells. This is Sertoli cells. It looks like this tree-like branching is present sometime. And these are the Spermatogonia, pale type A, dark type A, type B. Spermatogonias develop into spermatocytes after meiosis division, primary and secondary spermatocyte, and they metamorphose to form the spermatids. Spermatids are forming the spermatozoons. Okay. These are the ultrastructure of Sertoli cell and its relation with various germ cells. See here, this large cell is the Sertoli cell, okay, and there are tight junctions in between adjacent Sertoli cells, and these are the different germ cells. This is basal layer Spermatogonium. This is primary spermatocyte. Here are secondary spermatocyte. These are the spermatids, and these are the spermatozoa near the lumen of the seminiferous tubule. Okay, understood? So these are the locations of different types of cells. So seminiferous epithelium can also be given as a short note. You have to discuss all these cells, their locations, and practice a schematic diagram. Okay. Next we go to the duct system, okay, of the testis. Seminiferous tubules drain into straight tubules and then into the testis, and that drains the efferent ducts. From the seminiferous tubule, the sperms are going to straight tubules, from there into the retitestis, from here into efferent ductules, from there into the epididymis. Straight tubules are known as tubuli recti. Short terminal part of seminiferous tubule forms the straight tubules or tubuli recti, and their proximal part is lined by simple columnar cells and Sertoli cells, whereas distal part is lined by the simple cuboidal cells. Okay. Next we go to retitestis. It is a complex series of anastomosing tubules in the mediastinum. They are lined by low cuboidal or low columnar with a single flagellum. Efferent ductules, they collect the sperms from the retitestis. It consists of 12 to 15 ducts, and they are lined by simple ciliated columnar epithelium resting on thin lamina propria and circular smooth muscles. Okay. So we have discussed these ducts. This is the process of spermatogenesis. This part is spermatocytosis, here is the meiosis, this is spermiogenesis. Okay. So primordial germ cells develop into type A duct spermatogonia. Mitosis occurs type A pale spermatogonia from duct to pale. Then type A is developing into type B spermatogonia via mitosis. Then here meiosis occurring, reducing division. Okay. So from 2N you are getting 1N, haploid, diploid to haploid, into primary spermatocytes. Meiosis 1 is leading to 2 secondary spermatocytes. Meiosis 2 is occurring, 4 spermatids are formed. And they are leaving behind the residual bodies which are phagocytosed by the Sertoli cells and developing into spermatozoa through the spermiogenesis process. And spermiation is release of the sperm from the Sertoli cells. Okay. So these are the process of development from spermatid to development of mature spermatozoon. Residual body is left, acrosomal cap is forming. Okay, Golgi apparatus are fused with each other to form the acrosomal cap. Mitochondria are arranged around the middle piece of the sperm, and this from the centrioles they are extending in length and forming the tail of the sperm. Okay, so this is the maturation process of spermiogenesis. And this is the structure of a mature sperm. This part is known as head, this is neck. In the neck region lies the centriole. and here lies the nucleus and the acrosomal cap, and in the mid piece lies the around the axial skeleton. This is the mitochondria arranged in the middle piece, and here is one annulus is present. Here is some fibrous sheath in the tail, and tail is extending like this, and this is a cross section of the tail. Here you will get 9 peripheral doublets, okay, coarse fibrils, and the central doublet. Okay, so this is how one sperm is formed. This is the mature structure of a mature sperm. So this is low histology of testis section through the in low magnification view, and this is high magnification. Sections of seminiferous tubule, outer covering tunica albuginea. Here is the tunica vasculosa. You can see the section of blood vessel within the septa and also here. Okay, and in between this section of seminiferous tubules, these small cells you can see, these are the interstitial cells of the Leydig. Okay, and this helps in secretion of the testosterone. So here are the interstitial cells of Leydig, sections of blood vessels, sections of seminiferous tubule lined by the seminiferous epithelium, and near the lumen you see these sperms, okay, with tails. So this is the summarization of histology of testis. It has coverings and lobules. Coverings are tunica albuginea, tunica vasculosa, and these coverings are enfolding, giving septa to divide the testis into incomplete lobules. There are 1 to 3 seminiferous tubules in each lobule. They are having seminiferous epithelium, having these cells, spermatogenic cells. These are Spermatogonia, type A, dark, type A, pale, and type B. And spermatocytes are primary and secondary spermatocytes, and there are spermatids and spermatozoa. Okay. Sertoli cells, they are large pyramidal cells, supporting cells, and they removes the residual body. Interstitial or Leydig cell, they are polygonal cells with round nucleus, lie in clusters between the seminiferous tubules. Then there is straight tubules lined by the Sertoli cells, and retitestis lined by simple cuboidal to columnar cells. Okay. So you have to write down these 2 points in your histocopy for histology of testis. Sections of seminiferous tubules lined by Spermatogonia, different stages of spermatocytes and Sertoli cells, and interstitial cells of Leydig in between the tubules are seen. And draw this diagram in your histocopy. Okay. Sections of these seminiferous tubules, and in between the sections of tubules, these are the interstitial cells of Leydig, and some sections of blood vessels can also you can see, and this each of the section of seminiferous tubule lined by the seminiferous epithelium, Spermatogonia, spermatocytes, spermatids, and then sperms. Okay, and some larger nucleus are of the Sertoli cell. Here these are the Sertoli cells. Okay, so you draw this diagram in your histocopy. This diagram you take a picture and you draw this. So for spotting you write down those 2 identification points. Okay, only 1 minute time is given for spotting in your exams. Okay, so this is the anatomical. These are the lobules having those seminiferous tubules, retitestis, efferent ductules, epididymis, head, body, tail, leading to vas deferens. This is structure of mature sperm. Okay. These are the coverings has been shown here: nucleus, nuclear envelope, then acrosomal covering, plasma membrane, and this is middle piece surrounded by the mitochondria. Okay. Here will be annulus and rest of the tail of the sperm. Okay, like this you will see under microscope. Outer covering albuginea, vasculosa, septa dividing into lobules, sections of the seminiferous tubules, interstitial cells of Leydig. And this is the region of the retitestis. Okay, so these are sections of seminiferous tubules here in the region of retitestis, and here is the efferent ductile. Okay, it will look like this. Enlarged view of the seminiferous tubule, different cells. Okay, the larger nuclei for the Sertoli cell. Here is one Sertoli cell. These are the Sertoli cells, and these are the different stages of germ cells: Spermatogonia, spermatocyte, spermatid, and mature sperms. Okay, so different stages of germ cells have been shown here, and these are the larger nuclei of Sertoli cells. Like this you see under the microscope. Here you can understand this, identify those Sertoli cell nuclei like this. Okay, that's all. Any queries? Okay, so for testis, histology of testis itself is a important short note in your theory. Spermatogenesis process can come as a long question, and those terms are very, very important. Okay. Okay, so Sertoli cell, Leydig cell can individually come as a short note. So you prepare all these for your theories and practice a schematic diagram for your practicals. If it comes in theory, you also have to draw a diagram, histological picture of the testis. Okay, and prepare it for spotter. See the sections of seminiferous tubules. On upper this side, you can see the outermost covering, tunica, albuginea, vasculosa, like this. Are in between the sections of tubules, some areas are present connective tissue. Within those connective tissue, you can see the interstitial cells of Leydig. Okay, all those is visible here. Okay, see all this. And the sections of seminiferous tubules that always not be oval or rounded. It can be any kind of section, longitudinal section also you can get, cross section you can get, oval section, any type of section, because tubule is very much coiled. Okay, that's why so many see the multiple sections of the seminiferous tubules. And outer covering can be seen in this region, upper right corner, tunica, albuginea, vasculosa, and some septa, and within the vasculosa you see the sections of vessels. Okay, arteries and veins, sections of vessels you can see here. And you can see clearly the dots. Okay, some Sertoli cell nuclei in larger nuclei are of the Sertoli cells, and clear dots are seen for different layers of the spermatogenic epithelium. Okay, Spermatogonia, spermatids, spermatocytes. You cannot differentiate them, but you can see the different layers of nucleus. Okay, and near the lumen you can see the sperms with tail. Some of them are present.